Hi there, wishing a very good day to Professor Dr. Amir Faisal. First of all, thanks for being here. I'm Chong, who will be presenting my findings on the topic, the importance of non-coding DNA in relation to genome anatomy, mapping, evolution, analysis, and genome alteration. Based on the given topic, we can categorize it under 5 main aspects, which are genome anatomy, mapping, evolution, analysis, and genome alteration. Before we start up with our main topic, let's have a quick recap on what non-coding DNA is. Non-coding DNA sequences are components of an organism's DNA that do not encode protein sequences. They are historically referred to as junk DNA, and these non-coding regions are now recognized to serve other important functions. Some non-coding DNA is transcribed into functional non-coding RNA molecules in which the amount of non-coding DNA varies greatly among the species. Some examples of non-coding DNA are satellite DNA, telomeres, introns, cRNA genes, and gene regulatory sequences. Hence, without any further ado, let me proceed to my presentation by explaining the aspects accordingly. Firstly, let's have a look on our first aspect, which is genome anatomy. The non-coding DNA controls the gene activity. This is because they do not provide instructions for making proteins. Scientists once thought non-coding DNA was junk with unknown purpose. However, it was found that at least one of it is integral to the function of cells, particularly the control of gene activity. They help determine when and where certain genes are turned on or off, whereby it is an essential control for production of protein. To be precise, the non-coding DNA is also important for protein assembly, in which the genes are switched off, the process of transcription stops. This means that no mRNA is being made for the gene, and therefore no protein can be made. Next, let's move on to our second aspect, which is mapping. Linkage mapping often identifies chromosomal regions associated with a disease with no evidence of functional coding variants of genes within the regions, suggesting that the disease-causing genetic variance lies in non-coding DNA. The significance of non-coding DNA mutations in cancer was explored in April 2013. The non-coding genetic polymorphism plays a role in infectious disease susceptibility. For example, hepatitis C and also Ewing sarcoma, well known as an aggressive pediatric bone cancer. Some specific sequences of non-coding DNA may be features essential to chromosome structure, centromere function, and also recognition of homologous chromosomes during meiosis. Furthermore, let's proceed to our next aspect, which is evolution. The random non-coding sections of DNA can quickly evolve to produce new proteins. These de novo or from scratch genes provide a new, unexplored way that proteins evolve and contribute to biodiversity, according to the scientists. The non-coding changes are more likely than coding changes to be adaptive, because of a non-coding mutation is more likely to enhance a gene's function in one context without degrading it in other contexts, given that the gene expression in different contexts is often governed by distinct non-coding sequences. Whether seen as junk or not, non-coding DNA strongly boosts the shared genome size, thereby affecting a range of fitness-related phenotypic traits such as mutation rate, genomic flexibility, cell size, body size, morphology, growth rates, behavior, life cycle, and also speciation. As a consequence of less evolutionary pressure, non-coding regions of a gene allow for much more genetic variation than coding regions. This means that you will have more common and rare mutations in non-coding regions of a gene versus coding regions. Moreover, we can see the importance of non-coding DNA via the fourth aspect, which is analysis. According to the scientists, only about 1% of DNA is made up of protein-coded genes, and the other 99% are non-coding. The non-coding DNA does not provide instructions for making proteins, but it contains sequences that act as regulatory elements, determining when and where the genes are turned on and off. Some structural elements of chromosomes are also part of non-coding DNA. For example, the repeated non-coding DNA sequences at the end of chromosomes form telomeres. Telomeres protects the end of chromosomes from being degraded during the copying of genetic material. Besides, the repetitive non-coding DNA sequences also form satellite DNA. which is a part of other structural elements. Satellite DNA is the basis of the centromere, which is the constriction point of the X-shaped chromosome pair. The satellite DNA also forms heterochromatin, which is densely packed DNA that is important for controlling gene activity and maintaining the structure of chromosomes. The non-coding regions of DNA contain many of the markers used for identity testing. For example, short tandem repeats, also known as STR, are sequences of DNA that are 2 to 6 base pair long. The number of repeats in STR markers is highly variable among individuals, which makes STR useful in human identification. Lastly, let's have a look at the final aspect, which is genome alteration. The changes in genes can alter a protein's function in the body, causing health problems. Scientists have determined that the changes in non-coding DNA can also lead to disease. As mentioned earlier, the non-coding DNA controls gene activity, whereby it helps determine when and where the certain genes are turned on or off. Other regions of non-coding DNA are important for protein assembly too. If one of the regions in non-coding DNA is altered, a variant, also known as a mutation, in non-coding DNA can turn on a gene and cause a protein to be produced in the wrong place or at the wrong time. Alternatively, a variant can reduce or eliminate the production of an important protein when it is needed. Not all changes in non-coding DNA have an impact on health, but those that alter the pattern of a critical protein that can disrupt normal development or cause a health problem. For enhancer element, variants in non-coding DNA can disrupt other regulatory elements. These elements include promoters, where proteins that turn on gene attach, insulators, proteins that help shape the activity of genes in different ways attach, and silencers, where proteins that turn off gene attach. Some regions of non-coding DNA provide instructions for making certain RNA molecules that play roles in regulating genes activity or assembling proteins. Variants that interrupt these functional RNA molecules, such as transfer RNA, microRNAs, or non-coding RNAs, have also been implicated in disease. The same type of genetic changes that occurs in genes or that alter the structure of chromosomes can affect health and development when this occurs in non-coding DNA. These alterations include changes in single DNA building blocks, also known as substitution variants, insertions, deletions, duplications, and translocations. And do take note that the non-coding DNA variants can be inherited from a parent or acquired during a person's life. To sum up, non-coding DNA is important and it possesses crucial effects to many aspects in living organisms. Hence, I urge that more research should be done to explore more on non-coding DNA related to their functions. This is a list of reference related to my findings, and I would like to have an opportunity to thank Kok Heng, Bing Feng, and Sen Kai for sharing thoughts and ideas throughout the task. That's the end from my presentation. Thanks for watching and have a nice day.